In this video, we'll be looking at single point mutations in DNA. This is the DNA sequence we'll be looking at. In this DNA sequence, we are interested in a protein that is 4 amino acids long, so we'll have to find that first. In order to find this in particular, we'll need to transcript our DNA into RNA so that it can be translated into amino acids. When transcribing, it's important to remember that transcription is complementary and that mRNA is read from the 5' to 3' during translation. With this in mind, it's helpful to orient our DNA in the 3' to 5' direction. So let's look at this top strand first. Here's our top strand oriented from 3' to 5', and we can go ahead and transcribe that into our mRNA sequence here. In our mRNA sequence, we see AUG right here, which codes for methionine, our start codon. With our start codon found, we can move through our sequence to find the rest of our codons for this protein. So next we have valine, serine, lysine, and UAA, which is one of 3 stop codons. So we see that we have found our 4 amino acid protein code. Now we can look at our mutations. Here I've just isolated the coding sequence for our protein. For each mutated strand, we'll find where the point mutation occurs, whether it's a transversion or translation, and the effect of the mutation. We'll start with mutation A. Our original strand of DNA is underlined on top for comparison. Looking at the 2 strands, we can see that the mutated strand has a G here instead of a T. Because this mutation is from a pyrimidine to a purine, it's a transversion. To find the effect, we'll find what both codons code for. In our original strand, ATT transcribes to UAA, which is a stop codon. In our mutated strand, however, our mRNA codon becomes UCA, which codes for serine. This is called a read-through effect. Next, we'll move on to B. Here, our mutation is an A instead of a G, seen here. Since we're mutating from a purine to a purine, this is a translation. Once again, we'll find the amino acids both codons produce. In this instance, even though the third base is altered, both codons result in valine. For this reason, this is called a silent effect. Moving on to C, our mutation is a G instead of a T, which will be a transversion again. If we code our mRNA, we see this time we get a different amino acid from the mutation, arginine instead of serine, which we call a missense effect. Finally, let's look at D. Here, our T is mutated into an A, another transversion. If we transcribe and translate, we go from a lysine to a stop codon. This is known as a missense effect. And those are the 4 effects of single point mutations.